going to go on a quick tour through the digestive system. Take note of what happens at each stage and to each type of macromolecule. So we're going to start with ingestion in the mouth. In the mouth, we're going to begin a physical process, the physical process of chewing. Chewing also has another synonym we can use, mastication. And this is, like all physical processes, working to increase the surface area of the food. Increasing surface area means that there is greater availability of the substrate and the enzyme to meet each other, to do their job. So we chew in the mouth. We go down when we swallow. The epiglottis is pulled over top of the trachea. That covers up the trachea so food does not go into your lungs. Rather, it goes down the esophagus. It goes down through the process, as a physical process, of peristalsis. Peristalsis is the squeezing that happens one little bit after the next, one little bit after the next, so that the food material can go all the way down the esophagus past the esophageal sphincter. This is a thicker band of muscle right here that prevents stomach acid from coming back up. Once the esophageal sphincter is passed, the food enters the stomach, and in the stomach we have churning. So churning is just squishing, squeezing, rubbing. That happens just because the stomach is very muscular. It has those ridges, those bumps called rugae. I don't know how to draw them in here, so I didn't really do it, but the bumps on the inside of your stomach are called rugae. I'll just put that off to the side here. Maybe I'll draw some poor ones in there. You can maybe look at a better picture if you need to know. So after that churning, we also have, well, during the churning, we also have the excretion of gastric juice. We'll come back and talk about all the chemical stuff too. When the stomach is done, it's going to empty out through the next big band of muscle called the pyloric sphincter. The pyloric sphincter is the entryway into your small intestine. The first section of that is called the duodenum. In the duodenum, we're going to have stuff coming from both the liver and gallbladder. They work together. This is a team. And from the pancreas. From the liver and gallbladder, we're going to get bile. And bile will help with the job of emulsification. We're emulsifying, maybe I'll just write it out, just the lipids, not any other type of thing, just because the lipids are the only ones that are non-polar. They are hydrophobic. They don't mix well with water. We need to find a way to take one big blob and to break it into many, many smaller ones, and we do that by using bile, which is it's acting like a detergent here, in order to break that big blob into many smaller ones that are surrounded by a hydrophilic material, which is like the micelles we get when we use dish soap to clean up oils and fats. We continue down. The pancreas, of course, has a lot to do. We'll talk about that in the next video with the chemical stuff. We continue down the duodenum into the first section of the small intestine called the jejunum. In the jejunum and the ileum, we're going to have more of another type of physical process, and that physical process is not quite the same as peristalsis, although not too different. It is called segmentation. Segmentation is when you have something like peristalsis, but instead of it going just in one direction, it goes a little bit forward, a little bit backwards, a little bit forward, a little bit backwards, which helps to mix. So the food is going to travel through, at this point it's called chyme, the duodenum, the jejunum. It's twistier than I've drawn it because I'm not very good at drawing. And through the last section of the small intestine called the ileum. All of your small intestine has and the duodenum and, the jejunum and the ileum have a lot of these bumps. I don't know how to draw them very well, but these ridges all throughout. Those ridges also have little bumps on them, and those little bumps are called microvilli, and the bigger bumps are called villi. The villi are where things get absorbed into your blood. By the time we reach the end of the ileum, so the end of the ileum is somewhere around here, before we get into the large intestine, we will have absorbed all of our different types of macromolecule, or at least the subunits of those macromolecules. We will have absorbed from carbohydrates, we will have gone all the way down to, we'll do like this, monosaccharides. Carbohydrates start as polysaccharides. I won't have room to finish that, so I'll break it into two. Break down into smaller and smaller and smaller polysaccharides, eventually disaccharides, and then monosaccharides. The lipids will break down into a glycerol and 3 fatty acids. We're just going to write that out here as a triglyceride. Triglyceride has 1 glycerol, that's the head, and 3 fatty acids that can either be saturated or unsaturated, saturated or unsaturated with hydrogen. The nucleic acids will break down into a bunch of pieces, but the only ones we care about right now are called nucleotides. And the proteins are big, complex 3D shapes that will be snipped into smaller and smaller pieces, and those pieces will become, well, smaller polypeptides and smaller and smaller and smaller, until eventually we get to our end product, the amino acids. Amino acids are the building blocks of proteins, nucleotides are the building blocks of nucleic acids, triglycerides are the building blocks of lipids, monosaccharides are the building blocks of carbohydrates. The monosaccharides, nucleotides, and amino acids all get absorbed into the blood, in the villi. They go by active transport from inside this tube into your blood. You have blood vessels inside here called capillaries, and those things will go into your blood and head off to the liver, where the liver will package the glucose into glycogen and the nucleotides and amino acids into other things you need. The lipids, though, are the exception. Those do not go into the blood because they would clog up your arteries and your veins and your capillaries. So instead, they go into your lymph. These go into the lymphatic system, which is under lower pressure and has a bunch of stuff in there that we don't want to keep in the blood. The rest of that material will head, so the things you haven't absorbed yet in your jejunum or your ileum, will head past the entryway into your large intestine called the ileocecal valve. They won't go into the appendix, or at least most of them won't, because the appendix is just tiny, but they'll go up, over, down, do a little wiggle, and out. And in this time in the large intestine, a lot of water will be absorbed out. A lot of, well, not a lot, but some vitamins produced by bacteria that live in there, and some other water-soluble minerals will come along too. Everything that can't be used by your body is going to be defecated. It's going to leave out the end of the anal sphincter near the last section of your large intestine called the rectum. I'll come back for another video where we talk about the enzymes involved in these processes.